The muscle cells are the highly specialized cells which help in conversion of chemical energy to the mechanical energy. And when it is converted into mechanical energy, it helps us to perform the work. When we talk about skeletal muscle, which comprises approximately 40% of the total muscles, these muscles act on the skeleton and help in various functions, for example, maintenance of posture, locomotion, speech, and even respiration. So let's talk about these skeletal muscles in detail. Now, the average size of person muscle is determined generally by the hereditary and also by a hormone which is testosterone. But here we see a very famous Olympic winner in weightlifting is Mirabai Chanu, who has trained her muscle, and by the process of training there is increase or the hypertrophy of the existing muscle, maybe by 30 to 60%. So what causes this increase in muscle mass? The muscular hypertrophy, is it an increase in number or is it an increase in the size? The characteristic of the muscle is that muscles undergo hyperplasia. It is mainly by the increase in diameter rather than the fiber number. The number remains constant, the diameter can increase. However, if in few cases we see that increase in number occurs, it is because of the splitting of the muscle fiber from the center along the entire length to form the new fiber. So this increase is also accompanied by the increase in the enzymes, then glycogen store, the phosphagen system. Why? Because the capacities of both anaerobic and the aerobic metabolism are also increased with an increase in the diameter of the muscle. So when there is muscular hyperplasia, as we seen in a normal physiological condition, the question is, whenever there is a patient of muscular dystrophy, why don't we see this hyperplasia in these patients and why there is no recovery of the muscle fibers if they are damaged? The answer lies within development of the muscle fiber. The muscle fiber differentiation is completed around the time of the birth. They develop from the muscle cells, the primitive cells, which then are differentiated, and then no new fibers are formed from these myoblasts. So whenever our skeletal muscle fibers are destroyed after birth, they cannot be replaced by division of existing muscle fibers. However, it is said that whenever muscles have to be formed, it is from the undifferentiated cell, which are known by the name of satellite cells, which are present adjacent to the muscle fibers, and they undergo differentiation. And even if they undergo differentiation, as we may say in muscular dystrophy, this capacity is limited. So it will not be able to restore a severely damaged muscle to its full length. That's the cause why there is muscular dystrophy, and which is generally progressive in nature. The basic structure of the skeletal muscle, if we talk about it, each muscle is composed of muscle fiber and they are surrounded by a connective tissue layer, endomysium, which surround each of these fibers, and then they form the fasciculi, which is covered by perimysium, and then finally this perimysium is a place where the blood vessels and nerves are present, which supply the individual muscle fibers. Finally, these fasciculi are typically joined together to form muscle, and the connective tissue sheath which surrounds this muscle is known by the name of epimysium. So when at the end of the muscle, the connective tissue layers come together to form tendon, which is attached to the skeletal muscle. So what is the role of this connective tissue? Is that these connective tissue serve to transmit movement of actin and myosin molecule to the skeletal to bring about the effective movement. One. Second role is this connective tissue layer also contribute to the passive tension of the muscle, and therefore they prevent the damage to the muscle fiber as a result of overstretching or contraction, or it may be both. So talking about the gross organization, these muscle fibers are composed of these multinucleated cells which have a diameter ranging from maybe 10 to 100 micrometer. You can see they are arranged in form of fasciculus, and each fiber you can see is formed surrounded by a connective tissue sheath which is known by the name of sarcolemma. It is basically the same as the plasma membrane of any cell. Then inside it we have the sarcoplasm, and sarcoplasm is very rich in ions like potassium, magnesium and phosphate. And we can correlate this with crush injuries in a person. Whenever there is a crush injuries, there is extensive damage to the muscles in a person. What will happen? This potassium will come into the blood, and the level of potassium, if they are high in the blood, they can cause cardiac problem, and they can prolong the period of repolarization that may be fatal to a person. So this sarcoplasm also have tremendous amount of mitochondria which lie parallel to the myofibril, because they have to convert the chemical energy to the mechanical form of energy in the ATP to generate force, as we said earlier. Now each skeletal muscle fibers contain bundle of filament by the name of myofibril, which have a variable diameter and variable length. They are arranged in a form of a regular repeating pattern which is present in the myofibril because of presence of two filaments, that is thick filament made up of myosin, approximately 1500 molecules, and a thin filament which are double in number made up of actin. So this banding pattern is only seen in the polarized light, and this banding or the striation pattern is due to regular organization of actin and the myosin protein within entire fiber, and it is not just the fibril but the pattern is observed throughout the muscle. Here in this we can see the different bands, as you can see in this electron micrograph of the sarcomere, which is the picture present here, the light band, which is approximately 1 micrometer in length, and it is an area which is present between the two thick bands. You can very well see it between the two thick bands. We have this I band. Then A band, which is anisometropic to the polarized light, is just area which is present between the overlapping of the thin filaments. Then we have H zone. It is a lighter area in the middle. It is produced in the middle of the A band where the thin filaments are not present. It consists of only and only thick filament. And then we have Z line which is present and it bisects the I band. You can see here. It helps in the attachment of these I band, and they represent the repeating contractile unit in the skeletal muscles. The M line, you can find the M line. The M line is present in the center, and it is the M line, the dark line, which is evident in the center of the sarcomere, and it includes protein that appear to be critical for the organization and alignment of the thick filament of the sarcomere. So let us talk about the sarcomere and understand the different type of filaments which are present in the sarcomere. Here the brown is being represented by the thick myosin filament. The blue one is the thin filament, that is actin, and titin, which is the anchoring protein, is represented by this dotted red line. So when we talk about the sarcomere, which is acting as the structural and functional unit of the muscle, it is present between the two Z lines. Here we have the Z lines and it is present in between. Then we have this A band, as we said, which is anisometropic to the light, and it is mainly composed of the myosin filament. Then we have the I band, which is lighter, and then M line, which is present in the center of the H zone. It is mainly comprising of the tail region of the myosin filament, and these are the titin molecules which help in anchoring of these myosin filaments to the Z disc, and they maintains the elasticity of the muscle. Sarcomere is the structural and functional unit of the muscle. The ends of the actin filament are attached to the Z line, as you can see here, and these Z lines are composed of filamentous protein which are different from the contractile actin and myosin filaments, and they passes crosswise across the myofibril and also crosswise from myofibril to myofibril, attaching to each other. So each sarcomere consists of half of the I band, then A band, and again half of the I band. You can see here A band in totality, half I band coming from one direction and the other half coming from the other direction. The length of the sarcomere at resting is 2.5 micrometer. 1 micrometer is myosin filament and 1.5 being I band. So during contraction it will reduce further because they are pulled toward the center, and during relaxation the length is 3.5 micrometer. So the length of the sarcomere bears a significance. Why? Because it also determines the length tension relationship. If we have an optimal length of the sarcomere, as we can see in dotted regions of B and C, where the maximum contraction is there, maximum tension is there, the area B and C, you can see that interaction of actin and myosin fiber is maximum, the cross bridge cycle is maximum, and the overlapping is minimal. But if we move on to zone A, you can see the contraction is less. Why? Because there is considerable overlapping of these actin filament, as a result of which less number of sites are available for myosin interaction. And what happens in site D? Here you can see the tension is very less, or the contraction is less. Why? Because the actin have been pulled away from interacting with the myosin filament. Although myosin is there, although actin is there, but they are not interacting. No cross bridge cycles, hence no contraction is there. Now the different components of a sarcomere, if we want to classify, we can classify them into contractile protein. Here you can see the actin and the myosin, actin the green one and myosin are present in center as black one with their polar heads. Then regulatory protein, which are troponin and tropomyosin, which are again present on the thin filament. And beside that we have also the anchoring protein, which are titin. You can see these spring-like structures, titin. Then we have nebulin, which are present along the filament of thin filament, and then we have dystrophin. So let us study each of the component of the sarcomere. So first we talk about the thick filament. Here you can see the thick filament has a diameter that is twice of the thin filament. Its molecular weight is 4,80,000 approximately, and it is made up of 6 polypeptide chain. Here in the figure we can see that it has 2 heavy chains which are wound around each other in a helical manner, and we have a light chain which are present only in the region of head. The heavy chain, as we said, they wrap around each other to form the tail and the body or the backbone for the thick filament. The light chain combine with the terminal part of the heads to form this globular head. And these globular heads are responsible for interacting with the actin filament. Beside that, we have also hinge region which attaches these heads to the arm region. The myosin filament, the length is 1.6. The important characteristic of this myosin filament that it is twisted, as a result of which the heads do not lie only in one plane. They lie in all 3 planes, that is each pair of the cross bridge is displaced from the previous one by 120 degree. As a result, all of the myosin head can form cross bridges in all directions. So we have that advantage because of the arrangement of myosin head. So they can interact with all the actin filaments lying in all 6 directions. So this is myosin light chains which are present in the region of head. They are of 2 types, the essential light chain. Essential, why? Because they contain the ATPase activity and myosin binding ATPase site is there. And the regulatory light chain, which influence the kinetics of myosin and actin. They do not play a direct role in muscle contraction, but they have a regulatory role. So this is a diagram which may represent the myosin filament, which is shown in red color, and surrounding this we have 6 actin filament, and because of the steric arrangement of the myosin head, it is able to interact with all these actin filament to bring about the contraction. Then next contractile protein is the thin filament, which has a length of 1 micrometer. The F filament, as you can see in this diagram, it is a helical structure which is forming the backbone of the actin. The base is attached to the Z line, whereas the end projects between the space which is present between the myosin molecules. So what is this F actin? How is it made? It is basically made up of actin molecules, which may be 300 to 400 in number. They are basically G actin molecule, and each G actin molecule is a globular protein. G stands for globular. So this is basically a globular protein, and each one of them is attached to an ADP molecule. You can see these dots, they are the binding site for attachment of myosin. So these G actin molecules, when they combine together, they form this filamentous F actin, as you can see here, and the active site of 2 F actin here you can see the distance is 2.7 nanometer, giving it a staggered appearance so that it can interact with the myosin. So this forms a 6 to 7 nanometer long polymerized chain, which is double stranded in structure, known by the name of thin filament. Now when we talk about tropomyosin, you can see they are brown colored structures which are present in the form of a rod, and this rod is overlying the region which is meant for binding with the myosin. So under resting condition, this tropomyosin rod-like structure covers these sites which is present over the actin filament, and therefore prevent the interaction between the actin and the myosin. So whenever there is presence of calcium and availability of ATP, the tropomyosin will shift in configuration. It will shift and go in the groove, as the result of which there will be uncovering of the sites so the actin and myosin can interact. So it is basically a regulatory protein. Another regulatory protein is troponin. Troponin, here you can see it is of 3 forms: troponin C, T and I. They are loosely attached globular proteins, so they can easily detach from their site. So troponin C has an affinity for calcium, and when it is activated, it binds to 4 calcium ions, leading to the conformational change resulting in interaction of actin and myosin. Troponin T, you can see it has affinity for tropomyosin, and troponin I has affinity for actin. So why and how they play a regulatory role is that normally in resting condition, troponin C is not bound to the calcium and tropomyosin is covering the sites of actin. So when calcium is available, this troponin undergoes a conformational change, resulting in a conformational change in tropomyosin molecule, as a result of which it will move away from the sites that are myosin binding sites. It will uncover those sites, as a result of which actin myosin interaction is possible. So next, coming to the anchoring protein. The anchoring protein, you can see in this figure, the different type are tropomodulin. Tropomodulin present in the center, located at the end of the thin filament toward the center. It participates in setting the length of the thin filament. How much will be the length? Next is CAPZ and actinin. They anchor the thin filament to the Z line. So Z line is here, and here we have this CAPZ capping the thin filament. Then we have nebulin, which is inextensible here, and it helps to align actin into a thin filament. Titin, as we said earlier, it is a heavy molecule. The weight of titin is 3 million maybe, and it is present as a molecular spring, and it is responsible for maintaining the passive elasticity, and molecular weight is 3 million. As I said, it is very important in maintaining the passive tension in a muscle. So when we talk about Duchenne muscular dystrophy, the defect here is not in contractile, not in regulatory protein. The defect lies in the anchoring protein. Dystrophin, as you can see, is a large protein molecule. It is present on the intracellular surface of the sarcolemma, and when it is present with the integral membrane glycoprotein, it is known by the name of dystrophin glycoprotein complex. It is connecting the uh, membrane sarcolemma to these cytoskeletal structures, and therefore it maintains the structural link between the subsarcomal cytoskeletal element and the extracellular matrix. This helps to stabilize the sarcolemma. However, when there is a defect in this dystrophin, this is absent, so resulting in the damage to the sarcolemmal structure as such, and therefore the effects are seen. So this Duchenne muscular dystrophy is a genetic form of disorder. So whenever a child who is born and he starts walking, his milestones, for example, running, hopping, jumping, there are problem with the motor skill. So there is a problem. He will frequently fall. He will have trouble in getting up from the lying position or even climbing the stairs, which is also seen as Gower's sign. So shortness of breath will develop because respiratory muscles are involved. Fatigue will be there. Swelling of the feet may be due to the weakening of the heart muscle, resulting in characteristic edema. The problem of the breathing, as we said, respiratory muscles are involved. Gradually, the worsening of the muscles will occur. Lordosis will occur. Then the patient cannot walk. Weakness, especially in the pelvic muscles, and enlargement of calf muscles will be there. So all this will result, and this happens when a mother is carrier and father is normal. So this dystrophy may be manifest. The Duchenne muscular dystrophy can be diagnosed by either we take a biopsy from the muscle itself or estimates the enzyme, for example, creatine kinase, which is present in the muscle. That genetic test can be done to know the mutation that is affecting the gene, and electromyography can be used to study the various wave pattern in the muscle, and then we can compare it with the normal. So the treatment for Duchenne muscular dystrophy, no definite cure is possible, and it is usually seen that by the time the patient reaches the age of 25, he dies because of this lung disorder or respiratory problem. The treatment aimed is to control the symptoms and to improve the quality of life. The only hope that we have for this disease is that stem cell therapy has been tried in these patients, and it has offered some hope that these stem cells may be able to differentiate into muscle cells and help in the increasing the muscle size and number, and hence bring an improvement to these patients.